Okay, so hello everyone and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Professor Ilan Dinstein. Dr. Dinstein is an associate professor in the Psychology and Cognitive and Brain Sciences departments at Ben-Gurion University in Israel. He received his Ph.D. from the Center for Neural Science at NYU in 2010, and then completed his postdoctoral training in neurobiology department at the Weizmann Institute and the psychology department at Carnegie Mellon University. Since his arrival at BGU, he was awarded an Alon Fellowship, the Siretsky Award for Young Neuroscientists, and the Toronto Prize for Excellence in Research. Professor Dinstein has published over 45 papers in high-impact journals and is the director of the National Autism Research Center of Israel. I would just like to remind the audience to please address any question you have using the Q&A box or just raise your hand. Professor Dinstein, thank you very much for coming here today. The virtual stage is all yours.
Thanks all, and it's a pleasure to be here, and good evening from Israel. So I'll tell you a little bit about what I'm doing at BGU and then jump into this topic of neural variability that I hope you'll find interesting. I started my lab at BGU 10 years ago. We used to do a lot of neuroimaging. We're actually doing less neuroimaging as time goes by and are getting more heavily involved in using different technologies to track behavior. So we're doing a lot of computer vision and speech analysis and eye tracking and all sorts of combined measures of autism symptomatology with this major agenda of being able to measure outcome in children with autism. I'm actually going to tell you very little about this today, but I do want to say a couple of words about the National Autism Research Center in Israel. This is a big operation, so I have the pleasure of being of directing this operation. It's a community effort that we're together with Israeli government offices and the entire healthcare system. In Israel, there are 4 HMOs that give health services to the entire population. And so healthcare is very centralized. And this is a great opportunity to do autism research because we have access to a lot of data that is much more distributed in other places. And so a cornerstone of this effort is creating a national autism database. You can get a feeling here for the numbers. We're now up to about 1,200 kids with autism. We collect a wide variety of behavioral measures and record video and audio on many of them. And we're also doing some neuroimaging, some EEG during sleep, some sensors. We're now using like Fitbit trackers and things like that with some of the children. And the entire effort is dedicated to improving the diagnosis and the treatment of autism. So this is the focus is very translational in nature. And we this year received, it's just about to come out in the news in Israel, we received $12.5 million from the Azrieli Foundation to really further develop the center and turn it into a national network with multiple sites. We do a lot of recruitment of families during the very early stages of autism development. And if any of this is interesting to you, then I would strongly encourage you to go to our website. It's autismisrael.org, and you'll be able to see a lot of the different types of research that we do there. Some of it is also genetic and molecular biology and other things that that other partners are performing at the center. Okay, so that's the National Autism Center. But I thought that since this is more of a neuroimaging and neuroscience gathering, I thought that I would talk about variability. So this is a topic that I've been very interested in for probably a decade. And I want to take you through a little journey of what what we've done over this period of time to try and understand neural variability. And by variability, I'm talking about the moment-to-moment changes that happen in neural activity and how they might have an impact on many different aspects of our behavior and so forth. So to just start demonstrating that, here's an example, a raster plot of responses of a single neuron from area MT of a macaque monkey. And as you can see, just with your naked eye, each row here is a different trial during the experiment. And you see that the neuron is responding differently across trials, right? The timing of the spikes are different and the firing rate is different. On some of these trials, it's actually very difficult to see that the neuron has responded at all to the stimulus. And this is the case both when you look at single cell recordings and when you look at population activity with different techniques like EEG or MEG or LFP or any, right, pick your flavor. And here is another example from, this is an occipital electrode during the repeated presentation of the same visual stimulus, a checkerboard. You can see individual trials here in different colors and the mean in black, right? And in neuroscience, we typically compute the mean with this underlying logic that we're looking for the response that consistently, repeatedly is associated with the stimulus, and that these trial by trial changes or differences are not interesting, that this is some kind of noise, right? We think of it as, it could be measurement noise or biological noise or something that's not interesting. And this variability, I just wanted to also mention, it exists on every level of resolution or every physiological level that you can think of, from, all the way from molecular events such as a neurotransmitter attaching to a receptor, right? So the affinity of the receptor is going to depend on many different factors in the tissue, like the temperature, or maybe other things, right? So there's going to be variability there. There's going to be variability in the way the cell responds. So the resting membrane potential fluctuates all the time. So the cell is sometimes more prone to generating an action potential than other times. And all the way up to, if you think about neural populations and how neuromodulation like epinephrine, the epinephrine system, or serotonin and dopamine, they all act as kind of a gain control for the cortex, right? So, so there you would have large scale influences on the responses of large cortical areas and competition between neural networks like the intrinsic and extrinsic system. So I could go on and on with many other different examples. The point is that variability exists in all these levels. And perhaps the, so I mentioned before that in neuroscience, we usually, we have this dogma of looking at the mean and not thinking about this variability. And the big question, so why should we think about this variability? Does it have any impact on something that would interest us, like perception, cognition, development, clinical conditions? What value do this measuring variability have for us? And so over the last several years, we've looked at this from different angles, and I'll share some of the findings that we've had. We're, of course, not the only group working on this, but I'll focus more on our studies just for the sake of time. So the first area of relevance is probably perception. And that seems very, I assume that'll be intuitive for you if you're familiar with signal detection theory, right? This is one of the favorite theories in psychology, which describes that our ability to detect a stimulus or discriminate between two stimuli is a function of the strength of the signal of the stimulus and a function of how noisy that signal is. So, right, so if you think about these distributions that we're used to seeing in these situations, right, the signal determines the distance between those two distributions. The stronger the signal, the further away they are. And the other feature of the distribution is its width, right? The standard deviation. And so if the distribution is narrower, which means that the signal is less noisy, this distribution basically represents the strength of the signal across many trials, right? So on some trials, there's going to be some variability there above and below the mean. And in situations where that variability is small, the signal detection should be easy. That's the idea. And if we follow this logic, then larger neural variability should be detrimental for perception. So before I jump into describing the experiment to you, I have to tell you about one other phenomena which is relevant to our discussion, which is this issue of variability quenching. So it turns out that trial-by-trial variability, like what I showed you before in that raster plot and in the EEG traces, these trial-by-trial variability measures have a dramatic change following stimulus presentation. So trial-by-trial variability is much larger before we present the stimulus when the brain is sort of in a resting state, right? When the visual system is not processing anything. And then when a stimulus is presented about 150 to 200 milliseconds after stimulus presentation, this trial-by-trial variability drops dramatically by something like between 30 and 50%. And here on the left, I'm showing you an example of a dramatic example of one of our most beautiful subjects who had the largest quenching, where you can really see it by eye. And this quenching is apparent not only in our most beautiful subject, it's apparent throughout many experiments in multiple animal models, whether you look at LFP or spiking, or even membrane potential fluctuations. And a really nice paper that demonstrates all of this is the Churchill paper from 2010. They took data from, I think, about 20 experiments and showed how reproducible this phenomena is across multiple brain areas and different conditions. And so when we think about the impact or connecting perception to neural variability, we would need to do it to several different measures here, right? We would want to know how pre-stimulus variability impacts perception. How does this quenching, the degree or magnitude of quenching, how is that related to perception? And also the final absolute variability during actual stimulus processing. And so that's what we did in this study, where we asked our participants to perform a discrimination task. This was a contrast discrimination. So this was a 2-interval forced choice setup. They saw 2 Gabor patches. One of them was always at 100% contrast, the other was lower. And they had to tell us which one was higher. And then we could take all of the trials that had the full contrast stimulus and look at the responses there and measure this trial-by-trial variability, okay? So one of the nice features about these experiments is because you have 2 intervals, you can actually do sort of an internal replication and measure the variability separately for each stimulus interval and see if you get the same result. So that's what we did here. You can see the relative variance here is the quenching, the percentage of drop in trial-by-trial variability. Okay, and what we did is we quantified that for each subject, and then we wanted to see whether that was related to their perceptual performance. So we computed both the threshold, their contrast discrimination threshold, and we computed the entire psychometric function to estimate the slope the psychometric curve. And what this slope tells you is basically how stable the threshold is. So when the psychometric curve has a very steep slope, it means that there's, it's like a delta function, right? So there's a quick transition between stimulus strength where the subject is at chance to a stimulus strength where the subject is peaking at their best performance. And right, and if you have a shallow slope, it means that there are more contrast differences where the subject is marginally accurate at. And so we looked at both of these perceptual performance measures and correlated them with we started by looking at the magnitude of quenching. Okay, so each point here represents a single subject. The x-axis here is the magnitude of quenching. So the negative values here mean that the subjects are quenching more. It means that the difference from their resting state to the stimulus processing is larger. They're reducing variability to a larger degree. And what you can see here is that these subjects tend to have steeper slopes, so more stable perception, right? So this is a bit confusing. The top row is the first stimulus and the bottom row is the second stimulus. So for both stimulus intervals, the slope, the better slope belonged to those who quenched more, as did the lower thresholds. Okay, so you have better performance for larger quenching. If we look at the absolute variance, right, I told you that we not only want to look at the difference between pre and post, we want to also look at at what happened before stimulus presentation. We find this very interesting result that it's actually, there is some correlation with the pre-stimulus variability, such that those subjects that have larger trial-to-trial variability before the stimulus is presented, they tend to have the higher slopes. But you know, so here the correlations with the thresholds were weaker than what you saw a moment ago for quenching. And it's also interesting that we found nothing at all for the amount of variability in the post-stimulus section or period. Okay, and what this means, so taken together, this suggests that individuals who quench to a larger degree and start from a higher starting point, if you like, tend to have the best perception. Now, to validate these results, we ran another experiment using a totally different set of subjects and another protocol with motion perception. And here, instead of discrimination, we used the detection task, and we asked subjects to detect the movement of a set of dots, right? So we can control the percentage of dots that are moving coherently to the right or to the left. The lower the percentage of coherent dots, the more difficult this is. So that's what we're testing. We're lowering and lowering the coherence and looking for the threshold of the subjects. And when we do this, we see similar amounts of variability quenching to this stimulus, about 30%. And we see, you know, a weaker but still significant correlation between the threshold and the degree of quenching. So again, those subjects who tend to quench more are the ones who have the lower thresholds. And a non-significant relationship with slope, but at least it's in the same direction. So if we take this together, then it seems that across two experiments, we see this relationship that larger quenching is associated with higher perceptual sensitivity, better thresholds, and with more stable perception. Okay, so a question that you might have already thought about is where does attention fit into all this? So people have talked about attention in relationship to variability for quite some time now. And there's a theme that attention is supposed to do two things, right? If we're still in line with the signal detection theory principles, one thing that attention is supposed to do is to enhance your signal. So it's some sort of potentially like a gain control that increases the response to the stimulus you're seeking, perhaps like a location in space. And the other thing is that it reduces trial by trial variability, so narrows the distribution, right? And here you see an example of that from macaque V4 neurons from study by Marlene Cohen, where you can see that this, and there's something interesting here, that the responses, the part of the response that changes when the monkey attends the location where the stimulus is being presented, there's an increase in the amplitude of the firing rate about 150 milliseconds after stimulus presentation. And what you see here, the black is the attended line. This is basically the variability. It's the Fano factor, is the variability divided by the mean. So the variability is dropping to a lower, right, such that the Fano factor is lower than the unattended condition. And so we sought out to do something similar in humans with EEG. And we used a pretty similar setup where we had attentional cues where the subjects were presented with an arrow that pointed either left or right. And then they had a task to judge the orientation of these Gabor stimuli. And in 70, or I don't remember if it was 70 or 80% of the trials, the direction of the arrow was congruent with the location of the stimulus, right? So these were valid trials. And you can see that on the valid trials, the subjects were more accurate and a little bit faster. So this means that they were actually using the cues and doing what we were hoping they would do. As opposed to the neutral trials are those where you get a bidirectional arrow, and the invalid trials are those where the stimulus is presented on the other side. So they seem to be using the cues. This was one condition of the experiment. In the other condition that actually happened before this task, we just presented them with the cues without any without any judgment or anything. And the reason to show them just the arrows, just the cues, was that we needed a baseline for what the variability quenching looked like given this stimulus. And what we then did is we compared the degree of quenching, variability quenching during the control and the attended condition. Okay, so what you see in gray is the control condition. And you can see that throughout. I'm showing you here responses from the right occipital parietal electrodes and the left occipital parietal electrodes. You can see that when subjects are attending the stimulus, there's a larger degree of quenching right throughout in all these conditions. But what's interesting is the difference between the attendant condition and the control in relationship to which direction the arrow is pointing. So it's easiest to explain here in the left electrodes. You can see that the difference between the black and the gray curve on the left here is smaller than the difference between these two curves, right? And here we're looking at the left-hand electrodes, so left cortex, right? We would expect left cortex to do more quenching when the arrow is pointing right, when the stimulus is going to be in the right hemifield, the right visual field. And that's indeed what you see here. You see a lateralized response for this attentional effect, right? That it's stronger in the left electrode in comparison to the right. It's also the case for the top row, but it's a little, you need to actually do the computation to the statistical comparison to see it. And that's what I'm showing you here. This is the actual difference between the amount of quenching in both of those in both situations, right? So we're looking at the difference across conditions in the left electrode and the right electrode. And you can see that the lateralization index has an opposite direction, right? So we're looking at the difference between left and right here. And this tells us that that an intentional cue drives a reduction in trial-by-trial neural variability. Okay, and that's the first time it's been shown in humans. Right, the even more interesting question maybe is, to what degree does this differentiate individual subjects? And I think this is the nicest part of the paper, is that we found that there was a correlation between the degree of the attentional effect. So I need to describe that. So the attentional effect is the degree to which a subject benefits from allocating attention. It's the difference in the accuracy that that subject had between the valid and the invalid condition. So some of us are good at utilizing cues and attending the right spatial location. Other subjects are not as good. You see the subject here in red, there's almost no difference between the valid and invalid condition. They're basically not attending, they're not using the cue, or they're unable to use the cue, okay? It's not benefiting them. And as you can see, the subject here in blue is the one outlined here, okay, with a large attentional effect. That's an individual who is also quenching more. Okay, so this is the difference in variance in the control minus discrimination task. So this subject has a larger difference between the control and the intended task. It means that attention is driving more neural quenching in that subject, okay, as opposed to this subject outlined in red down here, who has a much lower difference, okay. And again, there's an internal reproduction here between the trials where the arrow was pointing left and the trials where the arrow was pointing right. Okay, so we get the same correlation, which is not trivial. And this suggests that larger quenching is necessary for higher attentional benefit, okay, or at least that there's a relationship between these two variables. Okay, so we looked at perceptual performance, we looked at attention, and the next topic we wanted to check was more kind of abstract cognitive abilities. So we started looking at, the standard psychology textbook tasks, and you have but we wanted to create a set of tasks that had increasing levels of complexity and cognitive demand. Okay, so we had our subjects do 4 different tasks that include a more or less passive visual stimulation. They just saw checkerboards. We had them do a choice reaction time, which in this task, each one of these 2 shapes is associated with a button. When they see a triangle, they need to press one button. When they see a circle, they press the other one. So it's pretty simple, but your main objective is not to get too bored. And then we did a go, no-go task with the same shapes, right? So one shape was the go cue and the other shape was the no-go cue. And then we did a 2-back task with Chinese letters that you see here. So they had a stream of these 4 Chinese letters in random order. And in trials where the current letter was the same as the one 2 trials back, they had to press a button. And we had another twist in this experiment. We ran the whole thing twice in 2 separate sessions, separated by a year. Okay, so we could also look at the reliability of some of these measures over extended periods of time. So here you see the amount of quenching that you get in response to each one of these 4 tasks. And I think it's already pretty interesting to see that the degree of quenching in visual cortex, in visual electrodes, is pretty similar when you're performing a 2-back task, which is very demanding, right, with Chinese letters, versus a choice reaction time task that's very simple. And yet you see similar levels of quenching. What you see here in the 2 lines are the traces for the data in the first session and then the other session 1 year later. Now, when we looked at correlation with a variety of measures from these cognitive tasks, we didn't find anything. And this was rather disappointing. So what you see here are correlation coefficients for the 3 different tasks, the 2-back, the go, no go, and the choice reaction time. None of the correlations were significant either for quenching or the absolute pre and post neural variability measures. This was true whether we were looking at the accuracy of the performance on these tasks, the mean reaction time, the speed, and even the reaction time variability, which was particularly disappointing because I was really expecting that variable neural activity would be related to variable response times, right? And at least when measured in visual cortex, it isn't. So we didn't find any correlates here. But the second part of this study, I think, was very exciting. Because we had these measures across such an extended period of time, over a year, we could look at how they correspond in individual subjects. And what we found were extremely strong correlations between the magnitude of all the variability measures during the first and second sessions. Okay, so an individual that had strong quenching during their first session of CRT also had strong quenching when they came back a year later. And this suggests that these measures are very reliable. They don't change over time. And not only were they reliable across sessions, they were also very highly correlated across tasks. So despite the differences in demand and so on, an individual that has high quenching in one situation, in one task, also exhibited large quenching in other tasks. So this suggests that that this magnitude of variability is really an individual trait, at least in adulthood, that each one of us sort of develops to a state where we have a certain amount of neural variability and exhibit a certain degree of variability quenching when we observe stimuli or process stimuli. Okay, so that's that summary. How much time do we have? Okay, we'll go through this briefly. So another angle that we wanted to examine regarding all of this was the potential mechanism or trying to think about what might be driving variability quenching. And we were thinking that it's related to, that it could be related to this phenomena of alpha desynchronization or alpha block. But surprisingly, these two areas of the literature were totally dissociated. No one was connecting the two. And so just to illustrate what I'm talking about, right, you could have a drop in trial by trial variability that would be caused by a reduction in the amplitude of oscillatory power across multiple trials. But you could also have a drop in trial by trial variability if you sync the phase across trials. So, right, so high phase coherence would create a drop in variability as well. So to distinguish between these two options, what we did is we took a data set from MEG. We actually checked this across multiple data sets, but I'll just show you this example. So this is a data set that we stole from a close friend and collaborator, from Tobias Donner in Hamburg University. And we wanted to look at the same issues using a MEG recording, because with MEG, supposedly you can get better access to those higher frequencies that are attenuated in EEG recordings. And so we took a data set of 23 subjects who had recordings from this from their MEG system, and we selected an experiment that was it contained a localizer from another motion-induced blindness study, which isn't relevant to our discussion. But what is relevant is that all the subjects had about 300 trials where they saw this rotating cross stimulus. Okay, so you get the impression that it's a rotating ball when you look at this. And there was a small Gabor stimulus in to the side, which was equivalent on all trials. So that didn't alter anything. We were looking at occipital and parietal sensors here from the MEG recording. And here you can get a feeling for the responses. So we were particularly interested in these changes in in spectral responses. And you can see the nice high frequency broadband responses here to the stimulus. And the nice thing about this setup is that there's sustained responses. The duration of the stimulus here was 750 milliseconds. And you can see that these high frequency responses do persist throughout the stimulus presentation. Down here, you see the lower frequencies, the alpha and the beta frequencies, they drop, right? So they're in blue. This is what's often called the alpha block or the alpha desynchronization. And then in the really low frequencies, the delta band, you see an initial increase followed by a decrease. And so the question, the reason we wanted to look at all these oscillations was to see how do these changes correspond to the variability quenching story, right? And so what we did, I hope you can see the yellow curve. This is the original time series when we run the variability analysis, the trial by trial variability. You see this quenching, it goes down here to about 25%. And then what we did is we reran the same variability analysis, but this time, instead of running it on the raw signal, we bandpass filtered the signal and ran the same analysis on spectral bands, right? And so when we do this on the delta band, we get an increase in variability followed by a decrease. That's the green curve. But take a look at what happens when we do it only on the signal from the alpha beta band, right? You see this immense quenching. And this is really where the power of the variability quenching comes from. It's this relationship to the drop in power in these two frequency bands. You see that in the gamma band, there's nothing at all. And to look at that from another perspective, we can also look at those individual differences. And you see this very strong correlation between the variability quenching here on the y-axis, right? So this is the degree of quenching, and this very strong correlation with the amount of power reduction in the alpha beta bands, nothing with the gamma band, and something, some correlation with the delta band. That's significant, but not as strong. So this tells us that there's a strong relationship with power, but we wanted to distinguish that from what was happening with phase locking, right? So here I'm showing you an intertrial phase coherence analysis. And you can see that phase coherence behaves differently from power. There's this transient initial response. It's more visible when you look at it in percent change values, right? So there's an initial increase in phase coherence, but then phase coherence basically drops to almost zero. And then there is another response at the offset when you turn off the stimulus. So the temporal dynamic here is very different from what you see for variability quenching that is sustained throughout the stimulus presentation period and a bit afterwards. And there was also no correlation between the degree of phase coherence and the amount of variability quenching in individual subjects, right? So what this tells us is that variability quenching is mostly associated and potentially driven by these reductions in oscillatory power, particularly in the alpha beta band, in those frequencies. Okay, I want to spend the last 5 minutes switching gears a little bit and tell you a little bit about autism and how autism was related to this whole story. So with our autism population, we don't have as nice of an EEG story to tell. We haven't gone into the depths that you've just seen with respect to autism, but there is a relationship there seems to be a relationship such that individuals with autism tend to have larger trial-by-trial variability. And this story started with just looking at behavior and seeing a lot of reports in the literature that behavioral variability is larger in autism. It's larger in multiple disorders, not just autism. And that there's this general theme in some of the clinical psychology literature that where they typically measure reaction time variability. Here you see an example showing ADHD subjects with larger reaction time variability than controls. There are multiple reports about this in autism, schizophrenia, and other disorders. And so this general theme that larger reaction time variability means that there's some problem with your brain, right, which is very non-specific. In autism, you see an association not only with reaction time variability, but also with saccade variability. So saccades that on average are just as accurate, but trial by trial variability is much larger. And also larger variability with pitch. So we're doing a lot of this now, looking at the variability of pitch over time, the ability to control speech. And so, you know, despite the fact that in those cognitive experiments, we didn't find a relationship between the EEG measures of variability and those reaction time measures, you know, it's if there's more variable behavior, it must be coming from some larger neural variability, right? So there must be an association somewhere. And so to look at that, we ran, this is 10 years ago, we ran an fMRI experiment where we took a group of high-functioning adults with autism and a control group and asked them to complete several sensory tasks. So we had tasks in the visual, the auditory, and the somatosensory domain. We presented them with air puffs, with tactile stimuli, and we looked at their fMRI responses, and specifically those responses in the areas that responded most strongly to each stimulus category. So we designed or identified regions of interest, specifically with the 200 voxels that responded most in each domain, which probably correspond to the primary cortices. We also looked at thalamic responses. And we quantified, so with fMRI, you don't see any of that quenching story that I told you about. You just, what we did here is we just looked at trial by trial variability in the peak response, in those ROIs. And when you do that and compare the autism and control groups, you see that the response amplitude is pretty much the same across the two populations, but the standard deviations are across trials are larger in the autism group. And once you have the response amplitude and trial by trial variability, you can compute a signal to noise ratio, right? And there you see these differences of about 20 to 25% lower SNR in the autism group. We did a nice trick here that I'll tell you about, and then we can finish, I guess. We wanted to, right, with fMRI, you have much better spatial resolution. So we wanted to try and separate variability that's local to each one of these sensory systems from variability that might be driven globally by, so just looking at the time course of the entire gray matter. So we computed what's sometimes called the global component, like a time course of the fluctuations of the entire cortex that are perhaps more likely to be driven by neuromodulation, arousal, changes in blood pressure, respiration, some things that might be interesting and some things that are probably not to our story. And then we can separate the two by regressing out that global component time course. So when you look at those global components at just their standard deviation, you see that it's pretty similar across groups. It's a little larger in the autism group, but regressing out those global time courses has very little effect on your mean responses. It has a large effect on your standard deviation across trials. You're actually cleaning up a lot of the variability. The local variability is reduced by about 20 to 25%, but still you see these differences across the two groups. So the autism group still exhibiting larger variability and lower signal to noise ratios. So that's the main finding here. There was, this was an older experiment. It's a small group. There were 13 individuals with autism and 13 controls here. You can see though that there is a relationship in the sense that an individual that shows low SNR in one modality also shows, this is variability, trial by trial variability. So an individual that has low variability in one sensory domain also has low variability in the others. And you get, again, this individual characteristic or individual trait. All right, I'm going to skip all this. Just tell you that there is some evidence from other studies as well regarding larger variability in individuals with autism. There are critical questions about what happens in kids. Very few studies with children, particularly younger children. There must be heterogeneity across individuals with autism. Autism is not one thing. It's naive to expect a single reproducible result from all individuals with autism. So stratification is obviously an issue for autism research. And well, I could say a lot more about autism, but we're running out of time. One last word, variability is, so far we've been talking about it as something detrimental for perception. You need to quench variability when you attend, right? And so it's as if variability is something negative, but variability is, of course, extremely important for learning. It's no accident that variability is much higher in younger humans and animals. And there's the beautiful work from the Olvexi lab with the finches demonstrating how important variability is for learning mature songs, right? And so there's this theme that when you're young, you need a lot of variability to encounter and make a lot of trial and errors in order to learn the right thing. and that as you mature, this variability decreases, and you in turn develop your expertise. So it might be interesting to think of autism as a situation, in some cases of autism, where this process is not happening properly and perhaps remains at a more immature state. And that could have something to do specifically with difficulties with social interaction, because social interaction is the thing we learn latest in life. It's the thing that takes the most amount of time to develop, is understanding these social nuances that we get good at only at very advanced ages. And I think that's most of what I wanted to tell you. Neural variability is interesting, might pertain to a lot of other topics that we haven't talked about, and this angle, might have a lot of value. So with that, I want to thank Ayelet and Ilan, the two amazing graduate students who did most of this work, and the wonderful collaborators. So thank you, and I'll be happy to answer some questions if we have a few minutes.
Thank you very much, Professor Dinstein, for this great talk. And as you said, we now move to the Q&A phase. So I'd just like to remind the audience to please use the Q&A box, the chat, or just raise your hand. And while people are typing, I'll take advantage of the opportunity to just say that I was really amazed by the correlation you presented along a full year between those subjects. And maybe I missed it, but how old were they?
So those subjects were university students.
I see.
So they were between probably 23 and 28, something like that.
And do you expect to get very different results if you look at teenagers or older population?
My expectation is that the two extremes of life, either early development or aging, late aging, you would see changes in these measures, and that you would see a drop, right? Or maybe increased quenching as an individual becomes more mature. Given the lessons from the finches, then you would expect to see some transition to a more stable and less variable state. But I'm not aware of any real research on this, so I think there's a lot to do there.
Got it. Thank you. Another question I have is regarding the autistic population. Do you have any plans, or do you expect that measurements of variability could be used for characterization or grading of autistic stages?
Yeah, so definitely. So that's I think one of the most interesting aspects of this. We already do it with eye tracking measures. So we have, I don't have it in this presentation, but you can actually look at, well, we also do it with just saccade to target tasks. So we have tricks where we get even 2- and 3-year-old children to look at targets that we present in between segments of movies, and we tend to get quite a lot of cooperation from them. And we can measure the accuracy of these saccades, for example, and we do see a strong correlation between saccade accuracy and some of the behavioral measures of autism. And we're trying to do the same thing also with kinematic measures of movements when we record the children with like Kinect cameras and even with regular video cameras. And so that's where some of this research is going.
I see, thanks. So as a follow-up, maybe not only to kids, but from your experience with different modalities to examine those measurements, who you think is most practical for the autistic population? You mentioned MEG and now the optical movements and EEG.
So I think EEG is the most practical, and now there are all these dry electrode EEG systems. We're just starting to look at some of them. I'm hoping that they would be, right, they're wireless and they don't have a lot of electrodes. It's usually like 12 or 18 electrodes, but some of these systems might be more accessible for work with 2- and 3-year-old children in a situation where they're watching TV or something like that. And so but we don't have any practical experience with that yet.
I see. So, okay, so thank you very much. And I would like to thank you once again for this great talk. And we're hoping to see you next time in person.